right, yeah, I would like to tell you something today about machine learning and mostly how machine learning is different when we're looking at understanding life systems. So most of us who have been working in the area of bioinformatics for a while know that the data that comes out is extremely complex, and we don't only want to make predictions, but typically we also want to learn how to understand the system that we're measuring. So from all that data, we don't only want to learn to make predictions, but also we want to actually learn how the system or the biological system works itself. And so I will show you some examples here throughout the talk, but first I will give you a very brief introduction to machine learning, just so that we have the same terminology for those of you who may not have a background in machine learning. All right, so when I am talking about machine learning, let's for now assume that we need quite a lot of data to train a machine learning model. So we have input data and we have output data. And in this case, the output data would be the predictions. So if you have a machine learning model, and I depict it here as a black box, we also will treat it mostly as a black box in this talk. It's this black box that needs to be trained. And so the first thing to train it is that you need to show it a lot of examples. And so you will show it both examples of input, of things that it wants to make the predictions on, as well as output, the predictions it wants to make. And so if we are in this setting, we are actually talking about supervised learning. So learning where we have data that already gives examples of correct predictions, and the model, the parameters that are in this model are going to be trained with this data to make the predictions themselves. All right, so we have features that are the input data and labels that are the truths and on which the model can be trained. Now, for example, if we take this into a real example, we can think, for example, about sequence alignments as input. So we may take sequence conservation patterns. So if each line here would be a protein sequence, and then you have multiple homologs here to generate a full sequence profile. That can be, for example, your input, and your output may, for example, be the prediction of secondary structure of a protein. So per amino acid, so here per column, effectively you would predict whether it's alpha helix or beta strand or coil. And so the training data would then be a lot of conservation patterns, and the output data on which you would train would be a lot of secondary structure annotations. Now, you may have heard about deep learning, and there's been huge advances in deep learning. And deep learning is not so different. It's effectively the same as what I showed before. You have still a black box model. It's just that black box model is more complex. And actually, it's made up of loads of little black boxes. So each layer here actually takes input and output, just as before. And so the next layer can take the output of the previous layer is input to the next layer. And that means you can train more and more very effectively variables that are inside these layers. And these layers themselves may have very different architectures, as we call them. We're not going into that now. But with this, you can effectively compute very complex rules about the data. So if there's a D here but not an E here, then this is probably so you can start reasoning. This model can start to learn and reason about the data. But you do need to have enough examples. And so if we have this deep learning, we can get a huge number of learnable weights in this machine learning model. But that also means that to train this huge number of weights, we also need a lot of data to actually train the model in the first place. All right, so first a very quick example that a lot of you may have heard about, AlphaFold. And so that is the same as I described it before. We have input data, we have sequence data, in this case sequence, but also conservation patterns, and the output would actually be a full protein structure. So slightly more complex than just specific classes of secondary structure, but actually a full 3D structure. But this is still supervised learning because the model was trained on known structures. Now, if you would look at more detail of the architecture, you see that the architecture of the deep learning is much more complex. I won't go into detail here. There's many other webinars from ISCB explaining AlphaFold in way more detail, so that's not the point here. But what I do want to stress is because, of course, this has been a huge leap in structural bioinformatics. So something we haven't been able to do for about 20 years in CASP, in the competition where this was tested. This was really an unsolved problem. Now we actually get quite decent protein structure predictions, specifically for single domain proteins that are quite reliable, and specifically if they're monomers. But if you look at detail in the architecture or in the papers, what has changed between AlphaFold 1, which was already good, but AlphaFold 2 really made a huge leap. They actually threw out a lot of the physics. So a lot of the physics in the model, for example, that two atoms are not allowed to clash, was actually thrown out of the model as an explicit constraint. So while the predictions are much better, I would argue that actually our understanding of the physical system hasn't necessarily improved at all. And so that also means that, for example, AlphaFold can come up with these kind of predictions, which are in no way compact. So this is not to some extent, so this is purely based on evolutionary data and possibly good templates. But of course, if this was put in as a monomer, this protein structure would not fold like this. It needs the other parts, the other chain there, the other molecule to actually fold into a stable structure. So there's very little physics directly in the model and also to be extracted from the model. All right, and then of course another large advance are the large language models, maybe better known as ChatGPT. And these are also pre-trained on very large data sets. And this is kind of, they say this is an autoregressive model. So it's also kind of supervised, but not quite on the task you want it for. So it's kind of, it's trained on a different task than what you will actually use it for in the end. And what it's used for is it's trying to predict the next word in the sequence. And so this is done on huge bodies of text. So take all the text you can find and then train it for as long as possible with as many parameters in your deep learning model as possible. And we're going up to trillions of optimizable weights in these models now. So a huge amount of data is needed, huge amount of computer power is needed as well. And why I want to explain quickly this is that because I want to contrast it with the kind of typical data problems we have in the life sciences, where we may not have either this amount of computer power nor this amount of data available, and where we also to some extent want to gain understanding on the systems. All right. So that brings me immediately to the next point, and that is when we're trying to learn things about life science data. So typically to kind of perform this kind of deep learning architectures, like these kind of multi-layer architectures, our data has the wrong dimensions. And that what that means is that what I just called input and output, we will actually have a huge amount of features. So for example, if you think about DNA sequencing data, huge amount of genes per sample, but the number of samples that we have may be very limited, for which we have true labels. So for example, if you think about, for example, classification of tumor samples, the number of samples for which we know if, for example, we have a disease progression in a patient or not, may be much smaller than the number of features. And this is actually a really difficult dimension to learn over. Another point that I already kind of mentioned is that we often do not have an intuitive understanding of the data. So for example, where text and images we can read easily, as humans, we don't really have intuitive understanding of DNA or protein sequences or even expression profiles. So that means that it's more difficult to develop the machine learning methodology in the first place because we can't immediately see if the predictions make sense. It also means that secondary, we don't don't only want to make predictions, but we also actually want to learn to understand the systems. Now, I want to just give you some examples out of our research group to go to see where we kind of run in these kind of problems, and also where you can partially solve these problems. So I will go through 3 examples, and then the fourth example will actually be presented by Uemi van Gils, who you saw in the introduction. And so the first problem we're going to look at is if we can predict if a genomic alteration has impact. And so this is work that was mostly done by a PhD student that works with me, Soufiane Lakbier. And so the question is, if we have a gene, and if that has a mutation or some kind of alteration, so it can be a point mutation, but it could also be a structural variant. So for example, a deletion in that gene, or maybe even a translocation of part of the gene, will that have an impact on the system? Now, this is specifically in cancer biology a very important question, because in tumors are known to be chromosomally unstable. So that means they require many mutations of all different types. So sometimes a lot of point mutations, but sometimes also a lot of structural variants. And so your entire genome is structured. But not all of these mutations will actually have an impact on the tumor biology. But we want to actually pick out which of these mutations do have an impact. So the question you're looking at is if we have a gene here and we have a wild type, and then we have a gene, the same gene in a different sample, and we have a mutation, does this have an impact? And now what we look at the same time is gene expression data. So you want to see if we can observe this in the gene expression data. But we're not looking at the same gene, so we're not looking only at gene A here in the gene expression data. Now we're actually looking at the entire system. And so our hypothesis is that actually an impactful event, and that is specifically true in tumor biology, that will have actually dysregulate the entire system a little bit. Maybe not hugely, but it will make so a problem in gene A will actually have a trickle-down effect on the entire system. And so that we assume that. And then we're actually asking, can we see that in the data? Now, the first way we tackled this problem was with machine learning. And now maybe you see, like, how can I tackle this with machine learning? We can actually set this problem up in a very similar way as I just explained, with input and output data. So here's your black box again. In this case, it says RF, but this is a machine learning model. In this case, it's a random forest model, hence the RF in the middle. And now we give as features, as input data, we give it gene expression data and samples. And as output, so as truth labels, we give it actually wild type or mutations. So for example, we take the P53 gene and we see if it has a mutation or not. And those that do not have a mutation are wild types. And those that do have a mutation are labeled as mutated. And so what we're asking is, based on this expression data for each sample, can we actually already predict whether this specific gene is mutated or not? And here we actually use the machine learning model to see if there's a signal in the data. So if we can predict this, so if we can predict whether it's wild type or mutation, that means there's actually apparently a signal in the data that corresponds to wild type for mutation. We may not exactly know or understand what it is, but it does mean that the expression data can actually divide between wild type and mutation of this specific gene that we're looking at. So let's see if that works. And so that's exactly what we did. And here we're looking at P53. And here the first thing what we're trying to predict is point mutation versus no point mutation. And this is a ROC curve. For those of you who have never seen this before, I will not explain it in detail. But what you do need to know is that if you're in the top left corner here, then you have a perfect predictor. So if you would go up like this and then like this, you have a perfect predictor. And if you would go straight over the diagonal here, you basically have a random model that has no signal at all. Now you can clearly see that for this particular prediction task, predicting whether there's a mutation in p53, we get a clear signal out. But it's not perfect. It's not right here, and we can actually make this stronger. So if we change the labels and we actually give more accurate labels, we can actually see that we can even predict this better based on the mutation data. So if we say like, okay, we have 2 chromosomes. If you have 1 point mutation and in the other gene you actually have an alteration, typically a deletion in p53, then you see that the model becomes even stronger. So that means that the expression data is even more strongly correlated with the status if we take this definition of the p53 mutation. And this is really nice. This model is incredibly predictive, so that means there's a very strong signal. This is also not a surprise because p53 is a well-known driver gene. So this is actually what we would expect for this gene. Now there's a downside to this way of solving the problem because we still need quite a lot of data. So we need about 50 examples of wild type and 50 examples of mutations to be able to make this course and to say, okay, does this gene have impact or not? So we made a second approach, and this is a statistical approach. And we do the same thing. So we take the RNA expression data, and now we do a huge differential expression analysis. So just something like we would take DSEC or HR, it actually works with both. And we just look at the p-values. So we look at the p-values of every single gene. So this would be the p-value distribution of every single gene, say of p53. Now, you may not know this about p-value distributions, but there's a very little knowledge that is very useful to have. So if you look at the p-value distribution of a large set of data, and there's no signal in that data, you would get a uniform distribution. So you would just have a flat line here between 0 and 1. If there's signal in your data set, so there is a differential signal, you will see a peak here at the left. And that means there's quite a large proportion of your data. It has actually got a p-value that is very low. And so this peak actually shows the signal in your data. And so we can actually count the size of this peak, or another way of saying it, we can actually count the fraction of the data that is inside this peak. And so that is actually the number of genes or the fraction of genes that changes differential expression if you do this over all the data. So we can do this. And now these are some of the examples that come out. So now, for example, if we look at BRAF, which is also a known driver gene, we see that actually if we look generally at non-coding or SNVs, we don't actually see here in a red that there is a huge signal. In fact, the gray points here are the signal of the peak or the size of the peak that you would expect in a random model on this data set. So this is sampled, so this is when there's no signal at all. But here you see that if there's specifically a missense mutation, then it's suddenly you get this huge peak in your p-value distribution. And so that means there's a really large proportion of genes, in this case almost 40%, that is differentially expressed when you have a missense mutation in your BRAF gene. And so that means that really this has a full systemic effect on your gene expression. And so we can see this, we can do this for a lot of the known drivers. Now, I will not go into more detail here. This will be presented on ISCB this year as well. And Soufiane Lakbir will actually present more of his results. And he will also show that we can use this very effectively to look at the impact of structural variants. All right. Then I want to show a second example. So the second example is a very different system, and we want to see if we can predict if proteins can be captured inside extracellular vesicles, or EVs. And this work is done by Dea Gogishvili and Katerina Bauri. They're also 2 PhD students that work with me. And they are also part of a large consortium, the Myriade Consortium, and the aim of that consortium is to find biomarkers for neurodegenerative disease. So, for example, for Alzheimer's and Parkinson's. And we want to know where these proteins are, and they need to be measured either in the blood or in the CSF. But the problem is the biomarkers may be hidden in these extracellular vesicles here. And so actually we would like to know, before we even start trying to develop a biomarker that we want to measure in these liquids, if we can even detect it, or that they're kind of hidden or trapped inside of one of these extracellular vesicles. And of course, we also want to understand why some proteins end up in extracellular vesicles. So we want to predict it, but we also want to understand why. Now the prediction problem here is as follows. So we take in a sequence, and we also take several features such as secondary structure and disorder and hydrophobicity. And then we try to predict if it ends up in an extracellular vesicle or not. So this is your input data. The output data is whether it's in an extracellular vesicle or not. Looking at more detail here, very similar as before, we have the input data for a lot of different proteins. And we try to train a model. And then the tricky thing here is the output data. So how do we actually know if a protein is inside an extracellular vesicle or not? So what we do is we go to existing databases, Vesiclopedia and Exocarta, but actually deciding whether something is an extracellular vesicle or not, or measuring it, is not trivial, because you have to filter out the extracellular vesicles. Now, experimentally, this is really quite difficult. And so we need to really make a very clean set, and we could actually only use data that was quite recent, because the protocols for extracting EVs have recently really been updated. But if we did that, and we also correct for a mass spectrometry bias, so we see actually that some proteins are much more easily detectable by mass spectrometry than others, and some of that is also predictable on the sequence data, so we need to correct for that. Then we can actually generate quite a nice model. So we get a positive set of proteins that have been found in extracellular vesicles, and then a negative set of proteins that are in the first place detectable by mass spectrometry. And so then we have a positive and a negative set, and then we can train the machine learning model. And so here, the first question was, can we actually do that? So is there actually a signal in the amino acid sequence that tells us if a protein is sorted into an extracellular vesicle or not? And so the first thing here you have for your ROC curve again, and we see that indeed we can predict this, so it's clearly better than going straight to the diagonal. You also see two different models here. One is based on all features, so this also, for example, includes experimental annotations in UniProt, and the light blue line is only on features that are directly predicted from the sequence. And you see the model is a little bit less good, but it still performs rather well. Now, the interesting thing is when we look at the underlying features, so what actually makes proteins end up or associated with extracellular vesicles. And we see here that a lot of the post-translational modifications, specifically some quite exotic ones like palmitoylation, are very important to predict whether a protein is likely to end up in an EV or not. Now we cannot only do that. So here we read out the features or the feature importance for the entire model. Now, when we're using SHAP values, we can actually go a level down as well. So you can also look per specific prediction, why a specific prediction has been made by the model. So here, for example, we have a ribosomal protein that is very strongly predicted to be in EVs. And we can actually see that that prediction has mostly been made because this palmitoylation site is there. And you can also see that actually a lot of the other post-translational modification sites are present in this protein, and therefore it has a very high chance of being associated with an EV. On the other hand, here we have another protein that is very unlikely to be associated with an extracellular vesicle, and we see that the majority of the post-translational modification sites are actually down in this case. So you can really look at the value and see which way, in which direction it is pushed. All right, so that is a little bit on explaining and also understanding why a model makes specific predictions. And here we have learned that some of these post-translational modifications are actually really important for the association with extracellular vesicles. Then another example that I want to show, this is really specific for life science data, as I explained about the problem of the data dimensions, that we often don't have enough examples to train on. And here I actually want to show you that you can still use deep learning model even if you have very little data, but we need to have more data on a related problem. And so how can you do this? So the idea is that we want to use new learning techniques, say deep learning. We have very much data, but we really have the wrong dimensions. So we have very few examples, very few labeled samples to train on. So the output, what I explained earlier in your prediction models. And we have a lot of features per sample. And so this is typically the data and the dimensions of the data that we work with in the life sciences. And so the question is can we still effectively learn over this? And this is a project that was done by Henriette Capel. And she was at the time a master student. She has now started her PhD in Oxford, actually, in my old group. And she is trying to predict here in the model the interface residues of proteins. So the question she's trying to predict here is if you have two proteins and they interact, she wants to predict which residues in the protein in the sequence actually form the interface. And we are agnostic of the binding partner, so we don't predict it pairwise. We really just ask which residues in the protein data banks over all structures are annotated as an interface, and so lie close to another protein in multimeric structures. Now, as you can see, we actually have very little data here. So we have a lot of sequence data, we have much less structure data, and even much less protein-protein interaction data. So you don't have so much data to learn on. So this is a nice model to try out this idea. Can we learn still effectively with deep learning models if we have little data? And what we use here is a multitask learning model. We actually used a existing architecture of a secondary structure prediction method. But the key trick here is not to use a single task, but we actually want to predict multiple tasks at the same time. So the task of interest here is the protein-protein interface prediction, but we at the same time also try to predict for each residue whether it's buried inside the protein or whether the amino acid is exposed to the outside. For each amino acid, we try to predict the absolute solvent accessibility. We try to predict the secondary structure as classified in 3 classes and as classified in 8 classes. So we have a lot of different points here, and you can imagine that these are important related tasks. So for example, if you want to be on the surface of a protein, you want to predict that, then that's of course also important information to know whether you're likely to be an interface protein, an interface amino acid or not. So so these questions or these tasks, these additional tasks are clearly related to the protein-protein interface prediction task. So we can do this, and to make it more technical, what you would do is you would encode this in the loss function. And the loss function is basically the penalty function on which the model is trained. Now, if you do this, what you can see is that we can make a reasonable model. So we have an AUC of about 75%. So 50 would be random, 100 would be a perfect model. And so we see that in this case, the dark red bar here is the model in which we have this multitask learning setup. So we have a bit of interface data, and we have more data on which it can co-learn in terms of other structural features, because we have much more annotation. But now the really interesting thing comes, this is if we actually, because this is more like a proof of principle, if we throw out even more data. So what if we reduce the amount of protein-protein interface annotated data even more? And so here we go even back to a 200th part of the data. So we take a factor 200 and reduce the interface data by that amount. So we really have very few data to learn over. And what you can now see is that only the model that has this multitask learning setup and that can still kind of learn some of its model parameters over some of the other tasks can actually effectively learn this. And you see that the performance drops a bit, but it doesn't become a random model, so you can still learn. Whereas the other task models that don't do this cannot perform on the data anymore and cannot learn the task anymore. So here, this actually means that there is hope. So even in the life sciences where you have these difficult dimensions, if we can learn over related tasks, we may still be able to solve problems and actually make specific models. All right, then the last example is the part that Joemi van Gils will present. And she will present the work that she did together with another PhD student, Deja Gogoshvili, on how sticky are proteins. Joemi, can I give the floor to you?
Yes, thank you, Sanne. So just for time efficiency, Sanne is going to keep doing the slides, so you might hear me say next a lot in the next few minutes. With the question how sticky our proteins are, one thing to mention first, so you've already heard Sanne mention, for example, protein-protein interactions. Now, typically, residues can be divided into hydrophobic and hydrophilic residues. And the hydrophilic residues, they interact well with the water, but the hydrophobic ones don't. So typically, these end up in the core of the protein in its 3D structure. But when they're on the outside, they typically have a function. So if you think of, for example, protein complexes, the interface tends to be more hydrophobic. Now, also on this slide, you see in green a very specific structure of a protein aggregate, which is called an amyloid fibril. And these fibrils, they aggregate in this very specific beta strand structure. And as you can see here, highlighted in yellow are all the hydrophobic residues. And so you can also see that a lot of hydrophobic residues here are at the core of this fibril. If we think about these amyloid fibrils, they aggregate and they usually are involved in different diseases and specifically brain diseases. So we were kind of wondering, okay, why do these aggregates happen in the brain so much? So on the next slide. Yes. So first what we've done is see, okay, we observed this hydrophobicity. How important is it for these fibrils? And so we've done a very nice combination where we have a theoretical model, which we combine with simulations and experiments to kind of understand what happens with these fibrils. And what we see is that, so typically if you think about protein structure or aggregate formation, then the stability of these proteins can be delineated into two components. One is called the enthalpy, which is kind of the interactions that the protein have with the different residues within the protein and also with the water. And the other one is the entropy, which is kind of the flexibility or like how much motion can this protein have. And what we have seen in this work is that the hydrophobicity is very important for this enthalpic part, so the interaction part. And actually, we could really explain certain physical behaviors of the fibril just with the hydrophobicity. Now, what's really nice in this work is that we, on the one hand, we have the experimental part, so we can really measure these on actual amyloid fibrils. But then we combine it with the simulations. And the nice thing about the simulations is that for every experiment that we do, we have the actual snapshots of the protein. So they can really give us mechanistic insight into what these structures look like and what they are doing when we are going through this. But so for this, the main takeaway is that the hydrophobicity is very important. Next slide. Yes. And so then what we wanted to do is that, because of course for these structures, 3D structures are very hard to determine of a protein. So what we wanted to see is, okay, can we then predict the hydrophobicity of a protein from sequence? So here, indeed, like Sanne has been explaining models. So here the model input would be the amino acid sequence of a protein. Then it goes into our machine learning model, which is a black box. And the output would be, okay, how large is the hydrophobic surface area? And now what's really important for training these models in this case is, okay, how do we actually define define what the surface area of a protein is. So if we go to the next slide. So the first thing we did is from known 3D structures, develop a tool to calculate the surface hydrophobicity. So basically what we start with on the left is the 3D structure of a protein. And here all the hydrophobic residues are indicated in red and all the hydrophilic residues are indicated in blue. And so what this tool does is that it puts a network on the surface of a protein structure, where you can see with all the little black dots on the surface. And if the dots are close enough, they get an edge to form the network. Now then, for each dot, we decide if it's closest to a hydrophobic residue, it gets a label hydrophobic. If it's closest to a hydrophilic residue, it gets a hydrophilic label. And then we can find all the largest interconnected regions that are hydrophobic, and then those are the hydrophobic patches. And so we use those to calculate the surface hydrophobicity. Now for that, I think it's on the next slide. Yes. So we have 3 measures of hydrophobicity. So here you see the hydrophobic residues in red and yellow and the hydrophilic ones in blue. Now the ones highlighted, so the total hydrophobic surface area is just the sum of all the hydrophobic residues. So those are all the red and all the orange dots. Then we have the relative hydrophobic surface area, which is the total hydrophobic surface area divided by the total surface area, so also including the hydrophilic residues. And then finally, we have the largest hydrophobic patch, which is just the area in red. Now, the largest hydrophobic patch is important because we see that that is often the surface area that's, for example, important for these protein-protein interactions or protein ligand binding. So if we go to the next slide. So we've trained models to actually predict these 3 values of surface hydrophobicity. And what you can see on the right, so like Sanne mentioned, the closer you get to the top left corner, the better your predictions are. And that the total and the relative hydrophobic surface area are relatively easy to predict. We have 3 different models here that I'm not really going into the details of. But what you can see in general is that the total and the relative hydrophobic surface area are relatively easy to predict, whereas the largest hydrophobic patch is a bit more difficult. But still better than random in our case. And so then the next question we had is, okay, how does this then relate to the human proteome? So if we go to the next one. Yes. So we used our prediction model to make predictions for all proteins in the human proteome. So we could all the UniProt sequences made the prediction. And then we wanted to relate this to expression. So for every protein we can look into the human protein atlas and see, okay, how much is this expressed in the human body? And so what you see in the figures on the left, so for each of the 3 measures, you see 2 violin plots, and one contains the 10% of proteins in the human proteome that has the lowest expression, and the high on the right that has the 10% of proteins with the highest expression values. And then you see the distribution of the hydrophobicity for each of the 3 measures on the y-axis. Now, what you can see indicated with the red circles is that for each of the 3 measures, in a region that is in the proteins that have the lowest expression values, there's a region that has very high surface hydrophobicity, which indicates that generally proteins with a very large hydrophobic surface area get a low expression, which makes sense because then that could be a mechanism of the human body to prevent these kind of aggregation of proteins to happen. Now, if we specifically delineate this into different tissue types, which you can see in the table on the right. So what we have done here is an enrichment test to see, okay, do proteins of certain associated with certain tissues have a higher or lower expression, higher or lower surface hydrophobicity than we expect. And so what you can see for the brain is that it's significantly higher for 2 out of the 3 measures, and also higher for this one, just not significant. And so the brain in and of itself is relatively very, very hydrophobic. in terms of the proteome. Whereas, for example, if you look at liver, which has a lower expression of hydrophobic surface hydrophobic proteins than you would expect. And so the brain just has a relatively larger amount of these hydrophobic proteins, which could, for example, be because the brain has a relatively high amount of membrane due to, for example, the shape of neurons, but also myelin, which is generally a very fatty substance. And so that could just make the brain more prone to these kind of diseases. And if we go to the next slide. Yeah, so in summary, we can actually do predict this hydrophobicity with reasonable accuracy. The proteins with a large amount of hydrophobic surface area are lowly expressed, probably to prevent protein aggregation, which could lead to disease. And the brain proteome is very hydrophobic in general. And so we can kind of use this combination of different methods, different tools to really gain insight into what goes on mechanistically, which I think is really cool. So I think with that, I give the floor back to Sanne.
All right, thank you, Yomi. All right, let's go to the next slide. So that also concludes this webinar on understanding AI or understanding with AI life science data. I want to get you to some key points. So one of the points I showed you for example, the extracellular vesicle example is that you need data to train your model on, and that you often need to extremely carefully select your data. So you need to very carefully, especially about your prediction labels, so the task of interest, you need to very well stratify that data and very carefully select it. And in fact, we often see that if you select the noise out, so if we actually, so say you have a region where you say, oh, we're not sure if this is really good data. If you leave that data out, your model actually becomes stronger than if you would throw the noisy data in. So selecting only the labels or the part of the labeled data where you're really certain about really helps to make a stronger model. The second point is that to gain insight into predictions, you can actually read out your models, especially if you have crafted features. So if you can look back at your features and then you can see which features are most important. And you can both do that at model level, so at the global level, but you can also do that per prediction at the instance level. Learning a task with very little labeled data annotations may actually be possible if there is more data on related tasks, and we can do that in a multitask setting. And lastly, if we actually want to gain understanding on the system itself, machine learning itself is not enough. So we actually need to combine bioinformatics workflows, for example, to make these very careful data selections together with machine learning, but also simulations to get mechanistic insight into the problem. And the best is, of course, if you can also go backward and forward with focused experiments, because in the end, the computational part can mostly build new hypotheses that you would then need to test again in experimental work. All right, so generally, I would like to kind of use this multitude of computational techniques, bioinformatics workflows, explicit simulations, and explainable machine learning. And from this, we generate hypotheses, and then we work very closely together with experimental collaborators who help us to set up new focused experiments. And then the models and the explainable machine learning may actually also help us with the data interpretation. All right, for this, I would like to thank all the PhD students that were involved, all the collaborators in the life sciences, and also collaborators in computer science. And of course, the group at the Vrije Universiteit who has helped with this. And after that, there's time for questions.
Wonderful. Thank you so much, Sanne and Yomi. So yeah, now we open the floor for questions. Please write your questions in the Q&A chat. And we have our first question here from Simon Bruce. Thank you both for the great talk. Where do you think the largest opportunities lie for the application of machine learning in the life sciences? Are there particular areas in the life sciences you think will benefit the most from machine learning in the future?
So the easiest answer to that is this is where you will see the quickest gain, is there where you have the most data. And so actually the fact that we could have something like AlphaFold, where already actually a huge gain has been made at this point, is because we had such good structured data. So, and such good competition. So without a protein data bank, which has been an initiative where you have really good structured data for years, and where experimentalists really sat together and say, we have to collect this in a similar way, even though the data itself is very complex. We come up with a way and give that kind of data in a very structured format and have a lot of examples that you can learn over. So basically those areas that have their data more in order, those will also be the areas on which you can learn more effectively over well annotated data sets with a lot of examples. They can be used in this kind of multitask setting. So if you have, even though maybe it may not be the exact problem that you're trying to learn, as long as it is a related problem, and we have that in a very structured way, and we have a lot of data for it, that will really push the other fields forward. So where do I see forward? So in the oncology field, we have now quite a lot of public data. In the structure field, in the functional field, we also have a lot of annotations. But for example, what we miss is a lot of kind of physics. So we don't have a lot of large databases where we actually have physical constants on proteins. So we don't know protein binding strengths in large quantities. We don't know protein folding stabilities in large quantities. So there, I think actually it will be much more difficult to kind of gain quickly on this wave of, even though there is maybe also what could in the end give us more understanding of the system. So I think kind of the quick fruit is there where you already have a lot of data, but then kind of adapt it to the question you actually want to ask. And that is still something that we're also in the process. So we can now actually predict structures, but maybe the structure itself will not, having the structure of a protein will not solve or create a new medicine, right? But then being able to apply that structure, and that is what you see start coming up now, maybe designing new structures to bind something specifically, finding a drug that can bind specifically in a pocket. So you'll see that the related tasks of the problems that are solvable, those will kind of speed up.
Great. We have another question from Nisha Hemandar Kumar. Amazing talk. Currently, what are the ways to compensate for data sets with the wrong dimensions?
Yeah, so I think it's also about picking your method well. So you could see, for example, very nicely in the where we predicted the impact. You could see that with the machine learning model, and that's a simple machine learning model, not a deep learning model, where we use random forest in that case. Also things like XGBoost would work quite well, or maybe just simple logistic regression, where we have about 50 samples, which is about the lowest you can go. So 50 examples of each. That is kind of the lowest you can still go. And you still have to be extremely careful in these cases with your validation. So you really should have cross-validation, and you really should have a hold apart test set of data to benchmark on. And the thing that's really tricky is not to touch that test set to, for example, try different model architectures on. So what you see is that a lot of people actually secretly still learn a little bit over the test set. And so you have to be extremely careful there. But if you do that, you can kind of go as long as 50-50 on with these samples. If you want to use deep learning, you really should go towards the 10,000s of examples, preferably millions. So that's and of course, the more data, the deeper and more complex you can make your models. If you go all the way to the other, so you maybe have 5 examples, like I showed you in the statistics case, we can still gain. So if you go back to classic statistics or statistical methods, we can maybe go as low as kind of 5 examples of each. So it's also about choosing your method very wisely. What we also now see from the machine learning field, so if you, for example, look at ChatGPT, is that if we pre-train, so that's actually similar to this multitask idea, but if we pre-train on these huge bodies of data, and then we can refine the model a bit to make it specific for a task of interest. So you see the same thing now happen with protein language models, where we have very large data models trained on protein sequences, trying to predict left out parts of the sequence. And there is a lot of data, as you saw. There's also a lot of sequence data. But then now we can then we get embeddings, as that's called, for each residue, and then we can we need much fewer data to retrain those embeddings for new specific tasks, because there's effectively already a lot of information in those embeddings. So it's the same idea again. If you can find related tasks that can help you to train a model, then you may still be in the game with more complex models, even if you have little data.
Can I add one to that?
Yes.
So one thing, and that's kind of specific also for biological data that you can do is to use kind of a biological validation. So for example, we had this other project where we actually had a very small data set. We had 3 groups and in total 26 samples, which is really, really minimal. But then what we did is that we used as a validation known interactions between different proteins, or maybe different protein pathways. So then you can check if either the predictions or clusters that you make sense from a biological perspective. So then you also get a little bit more back with the link with basically experimental work that has already been done. So if you have very little data, that could also be just like a mental check for, okay, does what I do actually make sense?
Great. I actually have another question for you, Yoami, related to your model on predicting sticky proteins. So you had you were doing quite well in predicting the more generic measures of hydrophobicity. When it came to the largest hydrophobic patch, that was a little bit more difficult to predict. Why do you think that is? How could you improve it? If you train your model only on the proteins that are lowly expressed, do you think you would get a better prediction for this largest hydrophobic patch? And would that make sense?
Yes, actually, that's a good question. Thank you. I think part of the reason we perform worse in this case is because the question in and of itself is a bit more difficult. So if you're trying to predict the relative or the total hydrophobic surface area, all you need to know is, okay, is this residue or part of this residue in the surface, yes or no? For the largest hydrophobic patch, you also need to know which of these proteins are connected to each other, which makes the task in and of itself a bit more difficult. So here, I think it would help to indeed select, for example, a data set where you very specifically know which is the largest hydrophobic patch, and maybe, for example, in this case, use something from protein-protein interaction interfaces or protein ligand binding, where you know exactly where the patch is, just because this problem in general is a bit more tricky.
Yeah, because you also need the topology effectively, right, of the protein.
Yes.
Great. And a question for Sanne regarding the first work you presented, which was looking at predicting impactful mutations. You're doing quite well on P53, but the P53 also has a really big impact, right, in the context of the cell. What about mutations that are less impactful? How much is your model able to predict the lesser effects for other proteins, other mutations?
So I think the interesting thing is we can still find new mutations that are impactful system-wide that have not been classified as such. So we see them popping up in the normal list of drivers, and they have similar high impact on the distribution. So in those we can find. The ones with a small impact, with a statistical method, you will not find, because basically, if that peak does not come over your uniform distribution, and actually because this data is so heterogeneous, you always have a small peak. So that's why we have a reference model for a specific data set. Then you will not see it. A machine learning model, on the other hand, would be able to see this, because it could also learn just on a few features, right? So it may only need 10 or 20 features to make a prediction either way, if it's strong. So say, for example, you would only see it in the expression of the gene itself and maybe one or two genes downstream from that, right? So that they're regulated by this. So it would be system-wide a very small effect, and we would, in theory, still be able to pick it up by the machine learning. In practice, though, you would need a lot of examples. So we could only do this for mutations that are relatively common, and so that means that roughly so if you, for example, take TCGA data sets, you would need to at least see it in about 5 to 10% of the patients. Otherwise, you could not get to these kind of numbers in order to train the machine learning.
Yeah, that's very interesting. I wonder, so thinking now of how much more we know about what's going on in cancer and pre-cancerous stages from recent studies done in normal tissues, where it turns out that the classical cancer drivers are really quite often mutated. I wonder if one could adapt your model to also kind of infer what's important, what's impactful in the normal tissue versus cancer, because there's the question of the drivers in cancer.
And we can even see this sometimes. So we see sometimes that if you, for example, look at this data, that in specific stages, because they have different stages, you actually see a larger effect, a higher impact effect than maybe in some of the later stages. And so if you have the corresponding data, so if you have normal samples and expression data for the normal samples, because that's not, you often don't have that, but if you do have it, you can ask this question without a problem. But what you see often is that you need a secondary mutation. So for example, you need also a deletion. So you don't only need a point mutation, but you actually need a deletion in addition to get really full impact on the system and to really become a strong driver to some extent. But these are really relevant questions, and also something we can now slowly start to get insight in.
Wonderful. Another question, so this is about the EV predictions. Are the EVs associated with degradation? Ubiquitination is often a marker for a protein to be degraded.
Yeah, that's definitely true. We don't actually really know. So I think that is, so the pomatoylation, that one was actually known to be associated. Ubiquitination, we're not sure, because it is sometimes part of the removal. So some of the proteins may be tagged for destruction, but that could actually be via the normal excretion pathway. It's not entirely clear why that pops up.
And regarding the impact of mutations, how much of this is a cause or a correlation between the expression and the mutation?
Yeah, that's always a difficult question, because of course we would like with the driver, we would like to see, and that actually also relates to your earlier question, you would actually like to see a cause, right, for the cancer. And with machine learning, the way we set it up now, you don't see causes. So what you can see, so if you use normal statistics, you can see kind of direct linear effects. If you look with machine learning, we can also see more complex effects. So this one on, that one off, that one on. So you can get more complex logic, but you cannot see causes. So in the end, these are just, so if we get a signal with the machine learning model, it still means there's a correlation between the expression levels and the mutation. So what we have seen in some very specific cases, if you have two subsets of cancer, so for example, for colorectal cancer, you may have MSI and MSS, and then you may also get different mutations that are known to be associated with those, and those can actually also cause expression levels. So we see this is actually rare. So we see the majority are actually known driver genes, but we've seen one or two cases where actually an existing split in the data that is somehow associated with a higher chance for a specific mutation also pops up. So we don't have a causal relationship in that way.